When you pour a glass of wine, it feels so straightforward, almost elemental. But here's something that might surprise you. Winemaking is actually a rather sophisticated biotechnological sequence.
It really is.
We're about to uncork the chemistry from grape to bottle, really digging into it for those of you curious about how complex processes actually work.
That's right. This deep dive takes us into the fascinating chemistry of winemaking and our exploration. It's based on a really detailed collection of research. It was presented way back at an American Chemical Society symposium in 1973.
Wow, 1973.
Yeah, it's remarkable, though, how much of that fundamental science still, underpins what happens in wineries today over half a century later.
Absolutely. So we've got these symposium proceedings, and they dig into everything, like the basic components of a grape, the wild transformations during fermentation,
Right, through to the subtleties of aging.
and even potential problems that can pop up. It's a real scientific goldmine, actually.
Our goal today really is to extract the crucial chemical shifts, the things that happen during winemaking. We want to give you a clear and hopefully engaging perspective on the science that, well, forms the basis of what many people just appreciate as an art.
Okay, so let's unpack this. Let's start right at the source.
Yeah.
The grape itself. The proceedings actually frame winemaking as this biotechnological sequence, and there's a helpful figure 1 in there that lays out the whole journey.
Yeah, figure 1. What's fascinating is how it maps out this whole progression. It starts with the grape's ripeness and its, overall condition.
Okay.
Then the tech steps, like crushing. Then the absolutely key part, yeast fermentation. Then you have the young wine stage, the various aging processes and the technologies they use, and finally, the wine that ends up in your bottle.
And each step influences the chemistry.
Profoundly. Each of these phases has a big influence on the chemical makeup we're going to discuss.
Definitely. Okay, so the most significant component of any grape and most fruits really is water.
Yeah.
It's huge, like 70 to 85%.
Yeah, it's the bulk of it.
Now, the amount of water clearly affects how much juice you can get. But it's more involved than just simple squeezing, isn't it?
Exactly. Think of it this way. That high water content means that all the other really interesting compounds we'll talk about, they start at pretty low concentrations.
Right.
So the later stages, fermentation and aging, they're all about concentrating and transforming these, sometimes trace amounts, into the complex flavors and aromas we associate with wine.
Oh.
And the juice yield itself, well, it's influenced by a whole range of things. The specific grape variety, how ripe it is, its general health.
Even the weather.
Oh, yeah, the climate where it grew, and of course, the types of equipment used for crushing and pressing. It's a real interaction of elements.
Makes sense.
And there's a particularly interesting case, Botrytis cinerea, the noble rod.
Oh, yeah, and sauternes and stuff?
Exactly, it shrivels the berries. This reduces the water content, but concentrates the sugars. That concentration is part of why those wines often command a higher price.
Talk about nature concentrating flavors. Now, speaking of concentration, let's get into the sugars. Glucose and fructose are the main players here, right?
Precisely. I mean, grapes do contain tiny amounts of other sugars, like stachylose and raffinose in Vitis vinifera, but yeah, glucose and fructose dominate.
And the ratio changes.
It does. What's interesting is how their ratio shifts as the grapes mature. Typically, glucose levels are higher when the grapes are unripe. And that ratio gets close to 1.0 as they reach maturity. Sometimes fructose even overtakes it if they get overripe.
This is where it gets really interesting. The symposium even gives specific examples. For a mature white Riesling, the glucose-fructose ratio was around 0.72.
Lower fructose, then.
Yeah. But in a Gamay Beaujolais, it was closer to 1.20, more glucose. And for some grapes grown in Ohio, this is interesting, they actually found significant levels of sucrose, unlike the vinifera grapes, where it's usually just a trace.
And that raises a really important point about how different grape species handle sugar accumulation. While Vitis vinifera primarily stores glucose and fructose, other commercially important species like Via labrusca, think Concord and Via rotundifolia, they can have quite significant amounts of sucrose. Sometimes as high as 6% in certain Ohio varieties mentioned.
6% sucrose, wow.
Yeah. And these subtle differences in how grapes store sugar, well, they have implications for how yeast metabolizes them during fermentation. It can lead to different aromatic compounds, influence the perceived sweetness, the body of the wine.
So like a higher fructose ratio might leave a bit more sweetness even if it ferments dry?
Potentially, yeah. Fructose is sweeter to our taste than glucose, so even small amounts of residual fructose can be perceived differently.
Okay, moving on to the tartness. The major organic acids in grapes are tartaric and malic acids. It's not just about the total amount of acid, but the balance between these two, isn't it?
Absolutely. The concentrations and that ratio of tartaric to malic acid, they vary quite a bit. Depends on the grape variety, how ripe it is. And interestingly, high temperatures and a lot of sunlight during ripening, they tend to decrease the overall acidity, specifically the levels of malic acid. Malic acid is more easily burnt off by the grape's metabolism in heat.
So the climate a grape is grown in really leaves its mark on the acidity profile.
Definitely.
And get this. The research highlighted that the main grape species in the U.S., Vitis vinifera, Vitis labrusca, and Vitis rotundifolia, along with a species from Afghanistan, are somewhat unique. Tartrates make up a larger proportion of their total acidity compared to mallets.
That's a key distinction.
The symposium even includes tables showing the total titratable acidity and pH for different grape varieties across a couple of years. Really demonstrates how much these things can vary season to season.
Variability is huge. Now let's think about amino acids. Their composition within grapes is, well, it's far from constant. It's a dynamic profile.
Influenced by lots of things.
Everything from the grape's maturity, the particular season, the specific location of the vineyard, the surrounding environmental conditions, it all plays a part.
Table 4 in the proceedings gives us a glimpse into the range of amino acid content in grapes. What struck me was the comparison with other fruits. Apples, for example, richer in things like asparagine, aspartic acid, glutamic acid, while pears contain more proline, serine, alanine, and both apples and pears share this unique amino acid 4-hydroxymethylproline. Never heard of that one.
It's quite specific, and what's fascinating here is that specific vineyard practices can also have a significant impact on these amino acid levels.
Like fertilizer.
Exactly. Table I shows that in Riesling grapes, the amount of amino acids, including nitrate levels and specific types of amino acids, is directly related to how much fertilizer was used.
And that matters for the yeast later on.
It really can. Yeast needs nitrogen, which it gets from these amino acids, for healthy fermentation. So vineyard practices can have this knock-on effect.
Right. And we can't overlook the phenolic compounds. Those are the real workhorses for flavor, color, and even texture, aren't they?
Absolutely. We're talking about things like leucoanthocyanins. Those are some of the compounds that give that slightly astringent, mouth-drying feeling, mainly found in seeds and stems.
And levels differ between grapes.
Yep, levels differ across varieties. And beyond those, you have a whole spectrum of other phenolics. Things like catechins and epicatechins, hydroxycinnamic acids like caffeic and shikimic acid,
Okay.
and flavanols like camphorol and quercetin. What's noteworthy is that in grapes, these flavanols often appear as monoglucosides and monoglucuronosides.
Meaning they're attached to a sugar.
Basically, yes. The flavanol molecule is attached to a sugar molecule, either glucose or glucuronic acid. This affects their solubility and how stable they are within the grape.
It's like each grape has its own intricate chemical signature, and it doesn't stop there. Grapes also release various hydrocarbon compounds, don't they? Little volatile things.
Absolutely. Researchers identified alkanes, alkenes, and aromatic hydrocarbons. They're present in that waxy layer on the grape skins. Mainly saturated hydrocarbons, C13 to C31 range.
Skin wax.
And let's not forget the inorganic components' essential minerals: potassium, calcium, iron, magnesium. They're all detailed in Table 6.
Important for yeast, too, I bet.
Very much so. Vitamins are also present and play a vital role for the yeast during fermentation, although generally, grapes contain sufficient amounts naturally.
Okay, so we've got this incredibly complex starting material in the grape.
Yeah.
Now comes the magic, or, well, the chemistry of fermentation. The yeasts are the ones that really kick things off, aren't they?
Precisely. Yeasts are the driving force. They consume those glucose and fructose sugars we just talked about.
The hexose sugars.
Right, and convert them into ethanol, the alcohol in wine and carbon dioxide. That's the fizz you see.
And in commercial winemaking, they don't just rely on whatever yeast is floating around.
Not usually, no. It's standard practice to use pure, cultivated strains of yeast. This helps ensure a clean, predictable fermentation. And it minimizes the risk of undesirable byproducts that could spoil the wine's quality. Off flavors, weird smells.
Got it. And very early on, often right after crushing, Sulfur dioxide, SO2, gets added. Seems like it has a few important jobs.
Yes, SO2 plays a really crucial role right at the start. Typically added around 100 to 125 milligrams per liter.
Okay.
It helps inhibit the growth of spoilage microorganisms, wild yeasts, bacteria that could cause problems.
Like an insurance policy.
Kind of. It also acts as an antioxidant, preventing enzymatic browning of the grape must, keeping it fresh looking. And interestingly, the yeasts themselves can even produce some SO2 during fermentation, sometimes up to 150 milligrams per liter.
Really? The yeast make it too?
They can, yeah. Depends on the strain and the conditions.
And for certain grapes, particularly those Native American varieties like Concord, I read that pectic enzymes are often necessary because they're sort of gummy.
That's a good way to put it.
Yeah.
Grapes like Concord have naturally high levels of pectin, which is a structural carbohydrate that makes things thick. Pectic enzymes are essential to break down these pectins. Well, it maximizes the amount of juice you can get from the grapes, and it makes the later steps, clarifying and filtering the wine, much, much easier. Less gunky.
Makes sense. Now, when we talk about red wine fermentation, it's distinctly different because of the skins, right? That's where the color comes from.
Exactly. Red wine fermentation happens while the grape juice is in contact with the skins, the alcohol that's produced during fermentation, and the carbon dioxide gas bubbling up.
Yeah.
They both act as solvents, helping to extract those anthocyanin pigments located in the skin.
So the fermentation itself helps pull out the color.
It does, and the skin contact also creates this floating mass of skins and pulp. It's known as the cap.
Right, I've heard of punching down the cap.
Exactly. Winemakers need to regularly submerge this cap through punching down or pumping over to ensure good color extraction and also prevent spoilage, because the cap exposed to air can grow nasty stuff.
So it's a balancing act. Getting color, but not too much. Bitterness.
It's a very delicate balance. Extracting enough color and desirable tannins without also extracting excessive amounts of harsh or bitter tannins. It's quite remarkable. Even the physical way the grape skin forms this cap actively contributes to the final chemistry of the color and structure.
And throughout this whole energetic process, keeping the temperature under control is vital for those yeast workers. Can't let them get too hot?
Absolutely critical. If the fermentation temperature gets too high, say around 40 to 45 degrees Celsius, which is like 104, 113 Fahrenheit, it can actually kill the yeast.
And stop the fermentation cold.
Exactly. Causes a stuck fermentation, which is obviously not what you want.
Okay, so fermentation's complete. We now have young wine. But the journey isn't over. Post-fermentation involves a lot of stabilization and, for many wines, aging. Let's start with tartrate stability. I've definitely seen those little crystals in a bottle sometimes.
Ah, yes, wine diamonds. What's fascinating is that newly fermented wines are often super saturated with potassium bitartrate.
Meaning there's more dissolved and should be stable.
Exactly. Like too much sugar dissolved in pulled tea eventually. Some will crystallize out to prevent these harmless but, visually unappealing crystals from forming in the bottle.
Because people might think something's wrong.
Right. Wineries commonly chill the wine, weigh down cold stabilization, or use ion exchange to remove the excess potassium bitartrate. Calcium can also form tartrate deposits, but the chilling usually takes care of that too.
Okay. And then there's protein stability. Cloudy wine is definitely less appealing.
Correct. Excess proteins in wine can cause haze or sedimentation, especially if the wine experiences temperature swings later on.
So how do they fix that?
To prevent this, winemakers use fining agents. Things like bentonite clay are common, but there are strict regulations. These agents need to remove the unstable proteins effectively.
But without messing up the wine.
Exactly, without altering the fundamental character of the wine or leaving behind any unwanted residues.
We also need to think about metal stability, specifically iron and copper. I remember reading about something called CAS. Sounds bad.
CAS refers to a type of cloudiness or haze. It can develop in wine because of colloidal complexes forming, involving metals, primarily iron and copper.
Where do they come from?
Well, the initial levels in grape juice are usually pretty low, but they can become problematic if they exceed certain thresholds, maybe 0.5 parts per million for copper, 10 ppm for iron, something like that.
And they cause problems besides cloudiness.
Oh, yeah. These metals can act as catalysts. They can speed up the rate at which white wines oxidize and turn brown. Interestingly, the yeast actually consumes some of these metals during fermentation, which helps lower the concentration.
That's handy.
It is.
Oh.
But excessive amounts often come from contact with winery equipment, brass fittings, old pumps, that kind of thing. Sometimes adding citric acid can help manage iron-induced clouds.
Right, and of course, microbial stability. Wine is tough, but some microbes can still survive and cause trouble.
That's right. Only a select group of microorganisms can really thrive in that high alcohol, low pH environment. Mainly certain yeasts, acetic acid bacteria, and lactic acid bacteria.
But no dangerous stuff.
Thankfully, no human pathogens can survive in wine. That's a big plus. The key is preventing contamination and controlling the growth of spoilage organisms.
No.
Using methods like careful management of SO2 levels, sometimes pasteurization, but that's rare for quality wine, and sterile filtration, especially right before bottling.
What kind of spoilage?
Well, undesirable yeasts like Cloacara or Hansenula can produce ethyl acetate. That gives a vinegary nail polish remover smell, a common fault.
Ugh.
Film-forming yeast can grow on the wine's surface if there's too much air exposure. And acetic acid bacteria, Acetobacter, they need oxygen to turn the ethanol into acetic acid, vinegar basically. So keeping air out is crucial.
Now, one post-fermentation process that sounds intentional, sometimes anyway, is malolactic fermentation, MLF. What's that about?
Malolactic fermentation, or MLF, it's a secondary fermentation. It's carried out by Leuconostoc bacteria mostly.
Not yeast this time.
Nope, bacteria. These bacteria convert the sharper-tasting malic acid, think green apples, into the softer, creamier lactic acid, think milk.
So it changes the feel of the wine.
It does. Reduces the overall acidity, adds complexity, often a buttery note, especially in Chardonnays. It often happens naturally, but winemakers can also kick it off by adding specific cultures like Leuconostoc Enos ML34.
Are there downsides?
Well, it can produce carbon dioxide, so maybe a slight spritz, sometimes temporary cloudiness or a slight color drop in reds. Occasionally, some off odors can pop up, but they usually blow off.
And you could test for it.
Yeah, interestingly, a relatively simple technique called paper chromatography can be used, even at home, to detect the presence of malic acid. Tells you if MLF is complete.
Cool. And then there's aging in wood. Oak barrels. It's not just storage, is it? The wood is doing things.
Oh, absolutely. The wood is an active participant. The ratio of the wine's surface area in contact with the wood to the total volume of wine is crucial.
Smaller barrels, more effect.
Exactly. Smaller barrels have a higher surface area to volume ratio, so a more pronounced effect. Oak contributes compounds, non-flavonoid compounds, phenolics to the wine, influencing aroma and flavor, vanilla, spice, toast notes.
Comes from the oak.
Largely, yes. And American oak versus European oak. They have different compositions. They contribute different amounts of extractable solids and phenols. European oak generally imparts more.
And the toasting matters, too.
Big time. The degree to which the barrels are toasted inside adds another layer of flavor complexity. Winemakers sometimes even pre-treat new barrels, leaching them to soften that intense new oak impact.
So many variables. And even the color of red wine keeps changing as it ages in barrel or bottle.
Yes, it evolves. Those vibrant red and purple anthocyanin pigments, they tend to decrease over time. Meanwhile, tannins, also from skins and seeds, and some from oak, play a bigger role in the color of older reds.
Turning more brick red.
Often, yeah. Contributing to those brick red or tawny hues you see in aged reds. Oxidation and condensation reactions happen between all these phenolic compounds, and the relationship between just measuring the anthocyanin level and the actual perceived color isn't always direct. Some forms become colorless, these flavines, which are unstable themselves. It's complex chemistry.
This is all incredibly detailed for Vitis vinifera, the classic European wine grapes. But the symposium also looked at winemaking from Native American grape varieties. How do they differ, chemically speaking?
Right. Native American grapes like Vitis labrusca, Concord, Vitis rotundifolia, muscadine, and various hybrids, they have some...
A chemical fingerprint. So these chemical differences mean you need specific winemaking techniques for Native American varieties.
Exactly. To deal with the naturally higher acidity and lower sugar, techniques like amelioration are often used.
Amelioration.
Basically, reducing the acidity, often by adding water, and sometimes adding sugar to boost the potential alcohol content. It's permitted for these grapes.
Okay.
Applying heat to the grape pulp can help extract color more effectively. And because of that higher pectin content, those pectic enzymes we mentioned, maybe used with cellulose fibers, are crucial for getting decent juice yield and pressability. Malolactic fermentation fermentation is also generally seen as beneficial for softening the acidity in these wines.
It's interesting to hear about the history, too. The symposium mentioned some early prominent native varieties like Cape or Alexander, Catawba, Isabella.
Yes, those play a really significant role in the early days of winemaking in America, especially the East Coast. Chemically, as one of the tables showed, native varieties generally have lower Brix levels, sugar content averaging around maybe 16 degrees, lower potential alcohol.
And the acidity.
Their Brix to acid ratios are also typically lower than vinifera. And as we said, that presence of diglucoside anthocyanins in Labrusca, Riparia, rupestris largely absent in vinifera, maybe trace amounts, that's a key chemical marker.
With all this intricate chemistry going on at every single stage, quality control must be absolutely paramount in winemaking. It's clearly not just, follow a recipe.
You've hit on a really critical point there. The quality of the raw material, the grapes themselves, is probably the single most fundamental factor, often more so than in other food production.
Really? More than like baking bread?
Arguably, yes, because you can't easily fix bad grapes. Even if you use identical winemaking techniques, differences in grape quality, which is heavily influenced by climate, especially the heat summation during the growing season, will lead to really noticeable differences in the final wine.
And quality isn't just something you check at the end, right? It's constant monitoring.
Absolutely. The symposium outlines key checkpoints all the way through, starting right in the vineyard, assessing maturity, condition, then through crushing and fermentation. Sanitation, temperature, yeast health, continues through processing and aging, regular checks on the wine in tanks or barrels, chemical analysis, sensory analysis, tasting constantly.
Tasting is analysis too.
Definitely. And finally, bottling requires careful checks. Incoming supplies like bottles and corks, sterile filtration if used, startup procedures. Table 2 in one chapter actually gives a really comprehensive list of all these QC points.
What kind of chemical tests?
Routine stuff like volatile acidity, checking for vinegar spoilage, SO2 levels, alcohol content, pH, total acidity, those are essential. Plus, specific tests for certain stages like measuring malic and lactic acid during MLF.
So it's a mix of lab work and tasting.
It really is a combination of scientific measurement and experience sensory evaluation at every single step.
It's almost like a continuous chemical and sensory audit of the whole process. Now, shifting gears slightly, the symposium also touches on home winemaking. The basic chemistry must be the same, but I imagine there are unique challenges on a smaller scale.
That's a great way to put it. At its core, yeah, home winemaking is the same fundamental chemistry, yeast turning sugar into alcohol and CO2. But the much smaller scale definitely introduces challenges.
Like what?
For instance, that surface area to volume ratio we talked about, it's much higher in small containers like carboys. That increases the risk of oxidation if you're not careful.
More air contact relative to the wine.
Exactly. Home winemakers often lack the fancy equipment wineries use for quick, low-oxygen processing or precise temperature control during fermentation. That can be tricky in a kitchen or basement.
Yeah, fermentation generates heat.
It does, and they usually have less direct control over the initial quality and consistency of the fruit they're using, whether it's grapes or other fruits.
And there are specific things home winemakers need to be particularly careful about during grape prep and fermentation.
Yes, definitely. It's important to avoid crushing the grape seeds if possible. They can release bitter compounds, sometimes a rancid flavor.
Okay, no seed crushing.
And the length of time the juice stays on the skins or pulp after crushing, that really affects flavor and tannin extraction. Careful tasting is key to decide when to press or separate.
How do they track fermentation?
Monitoring the progress using a hydrometer is very practical. It measures density or balling bricks. As sugar turns to alcohol, the density drops. It tells you how far along you are.
What about barrels at home?
Well, oak barrels can add nice complexity, but they can be really hard to manage effectively on a small scale, keeping them clean, topped up, preventing spoilage. It's tough. Many home winemakers stick to glass or stainless steel.
Simpler wines might be easier.
Often, yeah. Simpler dry white and red wines. Though many home winemakers get very creative with fruit wines, herb wines, even vegetable wines.
For reds at home, how do you judge the color tannin thing?
It's trickier without lab analysis. Visually assessing the color intensity is a common way, though less precise, to gauge when to separate the wine from the skins, trying to get good color without too much harsh tannin.
Can they check for MLF at home?
Yes, that paper chromatography method we mentioned, that's actually feasible for home winemakers to check if malic acid is gone.
And stabilization.
Proper stabilization usually involves racking siphoning the wine off the sediment multiple times and adding a small, carefully measured amount of SO2, usually as potassium metasulfite tablets, after fermentation is done. It helps protect the wine.
There's even mention of floor sherry. That's pretty advanced for home stuff.
It is quite specialized. That involves a specific yeast, Saccharomyces fermentati, forming a film, the floor, on the surface of the wine. It produces characteristic nutty aldehydic aromas by oxidizing some alcohol to acetaldehyde. Requires partially filled containers with air access. Definitely a trickier style to make successfully at home.
This whole process is incredibly involved, and it doesn't even stop with wine. The symposium also delves into distilling wines to make brandy. What are the key chemical points there?
Right, making brandy. Wines destined for brandy distillation have some specific requirements.
Different from table wine.
Yeah. Ideally, white grapes. Juice separated from the solids, the pomace, before fermentation. Crucially, no SO2 added during winemaking.
Why no SO2?
Because SO2 can lead to the formation of undesirable sulfur compounds and aldehydes during the heat of distillation. Also, a relatively cool fermentation temperature, maybe below 75 degrees F, 24 degrees C, is preferred.
To limit what?
To limit the formation of higher alcohols, the fusel oils, and distillation should ideally happen right after fermentation finishes, when the wine is fresh.
How is it distilled?
In California, continuous plate columns are common for brandy. They allow for separation of different components based on boiling points. A specific Heads fraction containing volatile aldehydes is removed early.
Get rid of the harsh stuff.
Exactly. Fusel oils, higher alcohols like amyl alcohol, are actually the most abundant congeners. Those are the flavor aroma compounds besides ethanol and water. Ethyl esters of fatty acids can also be significant, especially if yeast cells carry over into the still.
How do they get rid of the fusel oil then?
Specialized column designs and decanters are used to concentrate and remove these fusel oils, particularly the amyl alcohols, without letting the proof of the main distillate get too high, usually keeping it below 170 degrees proof initially.
And different compounds collect in different parts of the still.
Yeah, the distribution varies across the trays in the column. Higher alcohols and ethyl esters tend to concentrate on specific trays based on their different volatilities and the changing alcohol-water mix as you go up the column. There are figures showing this in the proceedings.
Fascinating. We already talked about oak for wine aging, but wooden containers must be crucial for brandy maturation too, right? Cognac, Armagnac. It's all about the wood.
Absolutely fundamental. The specific properties of the oak are critical. The way the wood is cut, quarter sawing is mentioned, helps minimize dimensional changes in the barrels as they age and soak up spirit.
And the wood structure itself.
Oak has these wide rays, medullary rays in its structure. They contribute significantly to its strength, its ability to be bent into staves, its stability, and its relative impermeability to liquid leaking out.
Different oaks matter here too.
Yes. Different species, like American white oak, Quercus alba, versus European oaks, Quercus robur, Quercus sessilis, they vary in the volume of these rays and their chemical composition.
So what does the oak do to the brandy?
As the brandy ages, the wood extracts various compounds into the spirit. Phenolics, tannins, lignins break down into vanillin. Lots of things. It also allows for very slow evaporation and oxygen exchange.
The angel share.
Exactly. And toasting the inside of the barrels before use is huge. It modifies the wood sugars and other compounds, creating those caramel, vanilla, spice, smoky notes.
And it depends on how long it's aged, the barrel history.
All those factors. American and European oak impart different flavors, with European oak often contributing more extractable solids and phenols. Preferences exist based on the source forest or region.
That surface area to volume thing again.
It still applies. Smaller barrels, faster maturation effect. You can even measure the increase in non-flavonoid content in the brandy as a rough indicator of how much wood aging it's seen.
Can they use alternatives like chips?
Yes. Oak alternatives like chips or staves can be used for flavoring, though often considered less complex than barrel aging. And just like with wine, new barrels for brandy are often treated or leached first to reduce that intense raw wood impact.
It's just incredible how much deep science underpins what seems like such a traditional, almost ancient craft. The symposium even gives a historical perspective on this.
That's right. Early winemaking evolved largely through trial and error. Regional preferences emerged using oak, sometimes resin for preservation or flavor.
Based on what worked or what they liked?
Yeah, exactly. It wasn't really until the last century or so that the fundamental biochemistry of fermentation became clear. thanks to pioneers like Lavoisier, Gay-Lussac, and then crucially, Louis Pasteur.
Pasteur figured out the yeast connection.
He definitively showed the essential role of yeast in fermentation, distinguishing it from spoilage. That knowledge was absolutely instrumental. It allowed winemakers to finally gain real control over the process, understand spoilage, and refine their techniques based on science, not just tradition.
So to sort of bring this all together, it's crystal clear that winemaking is so much more than just squishing grapes. It's this deeply fascinating, incredibly complex interplay of biology and chemistry.
It really is.
Starting with the grape's own unique, intricate composition, and then transforming through that remarkable dance of fermentation and the slow, gradual evolution during aging. And so many factors matter. The specific grape, the climate it grew in, the activity of those tiny yeast cells, the exact storage conditions. They all have a profound influence on the final character of what's in your glass.
Every step makes a difference.
For someone like you, listening, who appreciates the intricate workings of things, you can really see that even a seemingly simple pleasure, like a glass of wine, is underpinned by this incredibly complex biochemical ballet.
And what's truly intriguing to think about is how this deep chemical understanding ultimately informs both the art and the long-standing traditions of winemaking.
How so?
Well, it provides the scientific foundation for consistency, for making reliable wine year after year, but it also opens up exciting possibilities for innovation, for creating entirely new and distinctive styles of wine based on manipulating these chemical pathways.
Absolutely. And for you, our listener, thinking about all this chemistry, what specific aspects have really piqued your interest the most? What further questions do you now have about this amazing transformation from grape to bottle? There's a wealth of resources out there if you're interested in delving even deeper into the science behind your favorite drink.